So, the search for genetic material, even though the discovery of nuclein by Michel and the proposition for principles of inheritance by Mendel were almost at the same time, but that the DNA acts as a genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walton, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most of the cells. But the question of what molecule was actually the genetic material had not been answered. So let's read the transforming principle. So in 1928, the Frederick Griffith, in a series of experiments with Streptococcus pneumonia, is a bacterium that is responsible for causing pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism means bacteria had changed in physical form. When Streptococcus pneumococcus bacteria are grown on a culture plate, some produce smooth shiny colonies S while others produce rough colonies R. This is because the strain S bacteria have a mucus means polysaccharide coat, while R does not. Which was infected with the S strain, which was virulent, died from pneumonia infection, but mice which was infected with the R strain do not develop pneumonia. So S strain was injected into the mice, so mice died. R strain, when injected into the mice was alive. So Griffith was able to kill bacteria by heating them. He observed that heat killed S strain bacteria injected into the mice did not kill them. So S strain जिसको heat kill किए थे वो inject किया mice में तो mice was live. Now when S strain heat killed with R strain live was injected into the mice, then mice died. Means this was not effective, responsible for death, and this was also not, but still the mice died. So when he injected a mixture of heat killed S and live R bacteria, the mice died. So moreover, he recovered living S bacteria from the dead mice. वैसे तो उसने S को भी हीट किल कर दिया था, लेकिन जब डेड माइस में से रिकवर किया गया वो बैक्टीरिया, then it was live. So he concluded that the R strain bacteria had somehow been transformed by the heat kill S strain bacteria. Some transforming principle transferred from the heat kill S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. So this must be due to the transfer of the genetic material. However, the biochemical nature of genetic material was not defined from his experiments. It was biochemical characteristic of transforming principle was defined by the principles of from the experiments of Avery, MacLeod and McCarty. So prior to the work of Oswald Avery, Colin MacLeod and Macklin McCarty, 1933-43, the genetic material was thought to be a protein. So that time the genetic material was thought that it was a protein. So they worked to determine the biochemical nature of transforming principle in Griffith's experiment. So they purified biochemicals like protein, DNA and RNA from the heat killed S strain cells to see whether which one could transform the living R cells into S cells. So they discovered that DNA alone from S bacteria caused R bacteria to become transformed. So they also discovered that protein digesting enzymes like proteases and RNA digesting enzymes RNases did not affect the transformation. So the transforming substance was not a protein or RNA. Digestion with DNA is transformed did inhibit the transformation. So suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. So, can you think of any difference between DNAs and DNases? DNase is an enzyme, and DNase means many DNA. So, the genetic material is DNA. We have to prove in this. We have to study. Yeah. So the unequivocal proof that DNA is the genetic material came from the experiments of Alfred Hershey and Martha Chase in 1952. They worked with the viruses that infected the bacteria known as bacteriophage. So bacteriophage attached to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it is its own and subsequently manufactures more virus particles. So Hershey and Chase worked to discover whether it was protein or DNA from the virus that entered the bacteria. So they grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. So viruses which was grown in the presence of radioactive phosphorus contain radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not the radioactive DNA, because DNA does not contain sulfur. So here the radioactive phages were allowed to attach to E. coli bacteria. Then as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. So the virus particles were separated from the bacteria by spinning them in a centrifuge. So the bacteria which was infected with viruses that had radioactive DNA were radioactive. Okay, so the bacteria which was infected with viruses that had radioactive DNA were also radioactive. So indicating that DNA was the material that passed from the virus to the bacteria, and bacteria that was not infected with viruses that had radioactive proteins were not radioactive. So this indicates that proteins did not enter the bacteria from the viruses. So DNA is therefore the genetic material that is passed from the virus to bacteria. So this is the diagrammatic representation of Hershey and Chase experiment. Now the properties of genetic material, that is DNA versus RNA. We are just going to compare DNA and RNA. So from the foregoing discussion, it is clear that the debate between proteins versus DNA as the genetic material was unequivocally resolved from Hershey and Chase experiment. So it became an established fact that it is DNA that acts as a genetic material. So DNA acts as genetic material was proved by Hershey and Chase experiment. So however, it subsequently became clear that in some viruses, RNA is also the genetic material. RNA is the genetic material. DNA is not genetic material in RNA. In virus, sorry. So for example, in TMV and QB bacteriophage, okay, we found that RNA was the genetic material. So answer to some of the questions such as why DNA is the predominant genetic material, whereas RNA is just performs the dynamic function of messenger and adapter, has to be found from the differences between chemical structures of the two nucleic acid molecules. So let's dive into the structure. So can you recall the two chemical differences between DNA and RNA? Chemically, we are going to compare them. Okay, a molecule that can act as a genetic material must fulfill the following criteria. It should be able to generate its replica means replication करना चाहिए. It should be very chemically stable and structurally stable, and it should provide the scope for slow changes like mutation that are required for evolution, and it should be able to express itself in the form of Mendelian characters. So if one examines each experiment requirement one by one, because of the rule of base pairing and complementarity, then both the nucleic acids like DNA and RNA have the ability to direct their duplication. So the other molecules in the living system, such as proteins, fail to fulfill the first criteria itself. And the genetic material should be stable enough not to change with different stages of life cycle, age, or with change in physiology of the organism. Stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself. That heat which killed the bacteria at least did not destroy some of the properties of genetic material. So this implies that DNA is stable. So this now be explained can easily be explained in light of the DNA that the two strands being complementary, if separated by heating, come together when appropriate conditions are provided. So further, structurally, if we get into the chemical structural stability, okay. So in RNA, we can see that 2-OH group is present at every nucleotide in RNA, and it is a reactive group, and it makes what RNA is labile and easily degradable. So RNA is also known as what to be catalytic, hence reactive. So therefore, DNA is chemically is less reactive and structurally it is more stable when compared to RNA. Therefore, among the two nucleic acids, the DNA is a better genetic material. So in fact, the presence of thymine at the place of uracil also confers additional stability to DNA. So detailed discussion about this requires understanding of the process of repair of in DNA, and you will study these in higher classes. So both and RNA are able to mutate. In fact, RNA being unstable, mutate at a faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for the synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of proteins. The protein synthesizing machinery has evolved around RNA. So the above discussion indicates that both RNA and DNA can function as a genetic material, but DNA being more stable is preferred for storage of genetic information. For the transmission of genetic material, RNA is better.